I think I'll go ahead and get started. So welcome back, everyone. I want to start out here by reminding you that I much prefer these to be interactive. So if people are interested in talking, unmuting, and sharing video, I think it's more fun if we can have a conversation, but I also recognize that some people are in places that it's less comfortable to do that, and that's fine too. Today's lecture is really, or hopefully discussion, is going to be a continuation of what we talked about on Wednesday about genomics. And so once again, to remind you, we're talking about kind of the data acquisition aspect of this course, and starting with the genomics data. And so on Wednesday, we talked a lot about sequencing technology and just really DNA sequencing and genomes. And in today's lecture, what I'm going to focus on are different applications of that sequencing technology to really try to highlight the scope of the biology that can be learned using sequencing technologies. And there's far too many sequencing technologies to talk about everything today. But so the concept here is that we're going to kind of build off what we talked about on Wednesday and try to illustrate some cool applications and principles that I hope will kind of give you a sense of the scope, so that as you learn new technologies that we don't cover, you kind of have a sense of their context. Let's see. So before I go forward, though, I did want to just very briefly review what we talked about on Wednesday. And also because I rushed a little bit at the end, thankfully it was because we were having nice discussions earlier, but I didn't do much time for question and answer about the foundational sequencing technologies. So I have this slide here to remind me to just go back through. We talked about kind of the workflow of genomics experiments, starting from the isolation of the sample, which in the case of simple, genome sequencing is DNA that people isolate. We talked about some of the library preparation techniques, and while there are a huge number, we went in slightly greater depth into the standard library preparation for genomics with Illumina sequencing that had these Y-shaped adapters. And for some of you who know more advanced molecular biology, we had a chance to discuss why we did that a little bit. We talked about the sequencing by synthesis and the images that are created and interpreted, and how eventually those images, and the cycles from sequencing by synthesis lead to interpretation of not only A's, T's, C's, and G's, but also quality scores, and that those combined bits of information are put forward into analysis pipelines. And so just with that as a reminder of our last lecture, I want to pause for a moment here and ask if anyone has any questions about some of the sequencing technologies that we talked about on Wednesday. You should feel free to, go ahead, Catherine.
Yeah, do you mind just briefly explaining the significance for the Y adapters really quick?
Yeah, so this is a good question. So I think, let's see, I tried to set up, oopsie, go back here. So the key challenge, if you are making a library, let me see if this will work. I'm trying to be a little better today. Is this going to work? Oh, good. Okay, so here, if we have a piece of DNA, and we imagine that we were just going to ligate on an adapter that was a double-stranded adapter. So I'm going to draw it like this, where it's just a small piece of DNA, and I'm just emphasizing the 5' to 3' nature of it. We can call this A, and then if this is A, this entire sequence is going to be specified, because that's just a reverse complement if this is a double-stranded piece of DNA. And so essentially what that means is, if you have a piece of DNA, I'm just going to give it a little hook to illustrate which is a 5' and which is the 3'. If you take one of these guys and ligate it on, what will happen is you'll, of course, ligate in the correct direction here where you'll get A. And if this is double-stranded, of course, the reverse direction here because we would end up getting double-stranded stuff in both directions. What that means is that you would get A prime here, meaning the reverse complement. And so you've got a problem here, which is that the both directions of this DNA strand now have the exact same sequencing information at this side and that side, just reverse complemented from each other. And so if you did this, the problem would be if you started sequencing from this side, you would also end up starting to sequence from this side, because if you had a sequencing primer that started right here or a sequencing primer that started right there, they would be indistinguishable from each other. They would have the same sequence. And so this would create a problem because you would end up not being able to specify which of the two ends of your sequence you started from. If, on the other hand, we ligate on primers that look like this, then the concept here would be that you would get this Y shape, meaning that this guy here, I'm just going to call B, and this does not have to be B prime. This does not have to be the reverse complement because they're not complementary to each other. So I'll just call this C for a moment. So what that means, if we just follow this through, that means if that was A, this would be B. And if this is A prime, then we would end up with our C there. And what you'll notice now is that if we start sequencing from the C or C prime end, that's going to be a different end of the same DNA molecule than the B end. So whereas the normal ligation of a symmetric molecule would not distinguish the 5' and 3', these Y-shape adapters allow us to actually distinguish the 5' from the 3' of this molecule. Is that helping answer the question, Catherine?
Yeah, thank you.
Great. Let me clear those. Any more questions from last lecture before I move forward? All right. Well, clearly, feel free to interrupt if further questions come up. But so, oops, I'm still in the annotation, aren't I? Clear. Close that out. There we go. All right, so this is actually a slide from last time that I went through a little bit quickly, but just to kind of remind you the plot of the story as we went through all this work of how you get A's, T's, C's, and G's. And then kind of just in the last 5 to 10 minutes of the lecture, I kind of went quickly through this idea that, of course, just having a whole list of A's, T's, C's, and G's for a genome is not very useful in and of itself. And we really need context for those. And that context includes most simply genes, but also, you know, all the other types of things we might be interested in, like where, you know, in a cancer cell, we have differences from a non-cancer cell, where we have conservation across species, where the regulatory elements are, the control which genes are expressed, and that there's also information beyond what's encoded in the genome, that there's heritable information encoded in other levels, including the DNA methylation and chromatin modifications. And really, when we go through, these are the types of annotation we need in order to make sense out of the genome. And so you can think in one hand, this is just a problem of, you know, scientists and bioinformaticians and biologists going through and kind of looking at how to annotate the genome. And today, partly because it's the background of science that I'm interested in, and partly because I think it's a useful frame for this, I like to kind of step back and think about for a moment that the problem that we have of sorting through all the A's, T's, C's, and G's, in fact, is the same problem that your cells have. And so we can draw inspiration from biology and actually think about this from the cell's perspective of how does a cell figure out what pieces of DNA are relevant to it and what aren't. And so I just have this kind of provocative figure of, you know, thinking about how does a cell go through and use all the biomolecules that it's encoding in order to annotate its own molecules. And so I just want to remind you, I know that there's a diversity of backgrounds in this class in terms of biology. So I'm just going to go through very basic introduction to chromatin for those of you who already know this. Hopefully this will be a review, but please feel free to ask questions. So the idea, right, is that your DNA in your cell, once again, it's not just information stores in the microchip of a cell. It's instead, or in the hard disk of a cell, rather, it's a real chemical, and that chemical is not randomly distributed throughout the nucleus, but rather is a linear polymer that is wrapped around histones into chromatin. And this chromatin at its most basic form is can be divided up into individual nucleosomes, which are a set of proteins that are shown here in red, yellow, and blue. There, for those of you who remember this, the histones are H2, H2B in blue, H3 and H4 in yellow and red, together with the DNA that's shown in gray here. And this is a nucleosome shown here, and the nucleosome is a fundamental unit of chromatin. And it turns out that I'm telling you this not just to give you facts about how the DNA is packaged, but rather that really knowing a little bit about chromatin can help us understand how the cell annotates its own genome. And that information is very useful for also for us annotating genomics data. Okay, so what's particularly useful, of course, is not like if every single piece of DNA in the entire genome was packaged in the exact same way, then there wouldn't really be much information content there. And I would just say, oh, it's just a way to compress the DNA. But it turns out that these histones in the DNA can actually be covalently modified. And those are the modifications that I was talking about in the last slide. And for those of you like me who have a chemistry background, might appreciate actually looking at what these covalent modifications are, because they're simply chemical modifications. And the most important ones are very simple chemical modifications. They tend to be methylation events on the DNA or methylation events on lysine and arginine residues on the histones, or at times they're acetylation or phosphorylation events. And the idea is that depending on where you find these modifications, they mean different things to the cell. And so specifically what I mean by that is that you can think about where they are in a few different ways. You can think about where they are in the genome, meaning which nucleosome, what gene it's near. But you can also think about what locations are methylated. So for example, if a lysine is methylated, you can think about which of the histones has a lysine that's methylated, but even within each histone, there are many different lysines. And it turns out that depending on the location and degree of methylation, so lysines can be mono, di, or trimethylated, and they can be modified on many different residues. That the information about where it is and to what degree it's methylated actually contains information to the cell. And so people who are in the chromatin field end up using lots and lots of jargon associated with this, a few of which we'll go through today just for examples. For the purpose of genomics, I don't think it's necessarily important that you learn the specific modifications, but I think understanding what the language is and what it's referring to is important because it comes up enough in the genomics literature that I think it's worth learning, which is just that there's a handful of these modifications, and generally the way we talk about them is we just talk about which of the histones are modified. So remember, in the core nucleosome, there are 2 copies each of H2A, H2B, H3, and H4. So for example, if you have histone H3 that's modified, we just say H3, and then we'd say which residue. So if it's a lysine residue that's modified, which of the lysine residues. And so the histone sequences are such that there is a handful of lysine residues in the tail, and one that's particularly important for developmental biology is lysine 27. So we can talk about histone H3, K27, and then each lysine residue can get any of a number of modifications, including methylation and acetylation. And there are others as well, but these are the most common to come across. And so, for example, we can talk about histone H3, K27 acetylation. And for those of you who are aficionados of chromatin, you'll know that these different modifications mean different things and are frequently associated with different types of chromatin. And we'll go into that a little bit more. I just have this table, once again, not that it's important for you to learn these modifications, but I just want to kind of emphasize that different types of modifications are there beyond the ones I've drawn in this on this slide, including the ubiquitination, assimilation, which are small protein modifications. There are different amino acids that can be modified. There are different degrees of modification. And then we have abbreviations for each of those so that when we're talking about them, we don't have to spell all of it out. And so these are just examples of how you would go about looking up For example, is there an antibody that will bind specifically to histone H3 serine 10 phosphorylation? Well, this is a search term you need for PubMed if you wanted to look into that, or if you came across this, that's how you would parse that. Okay, so why am I bothering with this nomenclature? Well, the answer is that, this is in fact, pretty useful for thinking about how to annotate the genome. And the reason for that is, like I kind of hinted at already, if you look at the histones at different parts of the genome, they have different modifications on them. And those different modifications can tell you what type of content is in that region. So just to give you a few examples, so if you imagine right here, we have a gene that's being encoded. So let's imagine this is a protein coding gene. At most genes that are transcribed by RNA polymerase II, which I'll remind those of you who are a little further from your biology, RNA polymerase II is the major RNA polymerase that takes a DNA and turns it into RNA in transcription for the RNAs that will go on to encode for proteins. And pretty much these RNA pol2 genes have very stereotyped histone modifications right at their very beginning, including H3K4 trimethylation. And it turns out that's so good that if you just give me a map of all the places where you find histone H3K4 methylation, I can pretty much go through and find all the places that the polymerase is going to start transcribing throughout the genome. So it's kind of amazing, right? You can use this chemical modification and the cell is already annotated for us where the starts of the genes are. And of course, we have other ways to validate those and make sure they're true. And there are a few exceptions like everything in biology, but overall, it's an amazingly predictive mark for telling us where the 5' end of a gene is. And once again, my point here is not to show you every mark that's important, and the list kind of changes over time of where people think the most information is. But the idea is you get the starts of genes. You also can mark the entire open reading frame with modifications such as H3K36 trimethylation. There are other regulatory regions, including enhancers that turn on nearby genes, and those have different modifications, including H3K27 acetylation, which just happens to be the modification that I used as my example when we were talking about these modifications. And the idea is that together between the histone modifications, the transcription factors such as CTCF, which are proteins that recognize specific sequences of DNA, and DNA methylation, between those sets of information, the cell can really go through and figure out what each piece of the genome is used for. And of course, it's circular. Each one of those has to be replicated, and each one is regulated in various ways so that cells can change their identity over time. And since this isn't a chromatin lecture, I'm going to leave it at that, except to say that when we map these different modifications, we can learn a lot about the genes in any given cell, learn which ones are on because they have modifications that correlate with activity. There are other modifications which are associated with repressed chromatin in different cells, and there are different flavors of that. So the more you learn about the chromatin modifications, the more you can learn about the genome in general and the specific regulated state of that genome in the cell line that you're studying, or the cell that you're studying if you're doing single cell work. It's also worth emphasizing, I think it was kind of implicit in what I said in the last slide, but that, of course, different cells require different parts of their genome to be on at different times. And so therefore, if you're studying brain or limb, different genes are going to be on to make a limb or a brain. And even sometimes the exact same gene will be on, but different pieces of regulatory DNA will be responsible for turning on different genes in different tissues. And so these are just some of the complexities that are worth being aware of. And of course, if we want to make good use of genomics data, we want to be able to measure and take all of these different types of things into account. And so there are lots of techniques to study these modifications, where they bind, how the genome loops, and of course, ultimately how the RNA is made from these. And I'm not going to have time to go through all of them, but I just wanted to try to figure out a subset that gave us a range of the types of experiments that people want to know. and also give you a sense of different types of sequencing technologies out there. So I tried to simplify this down. I've learned that over Zoom it's a little bit harder to get through everything in the same amount of time. So I tried to cut down from what I included last time. But so here are just 3 examples of types of questions one might have, such as where are the regulatory elements? So if we're going to care about all the things that turn on and off different genes, how are we going to find where are those important pieces of DNA that are important for that? And I'll say a thing or two more about that in a moment here. But then, of course, I just went through that we care a lot about the chromatin composition, meaning where the transcription factors bind, where the histone modifications are, things like that. And then, of course, we also care about the RNA that gets made from the genome. And so we'll talk more about that as well. But I want to kind of break these up into a couple different, somewhat arbitrary categories. And these are mostly mine. This isn't something very important to distinguish, but I want to really emphasize that sometimes we know what we're looking for. You know, we have specific hypotheses about what factors are involved in something. And in that case, of course, we take targeted approaches to study those factors. But frequently, we're in a position where we actually don't know much about the cells we're studying, and we don't know which specific proteins or modifications are most important. And so there are a subset of global approaches that allow us to ask kind of more generic or fishing expedition type questions, to ask things such as, in all of my genome, where are there proteins that are bound to it, regardless of what they are? And then other times we want to take targeted approaches to only look at specific ones. So let's start with some of the global approaches, and let's think about the problem of the genome. And so I think many of you might already kind of anticipate that based on just a genetic code alone, you can actually use computational approaches to look for potential open reading frames that might encode for proteins, because we know the genetic code and we know what the stop codons are, and we can go through and we can figure out which regions of the genome have coding content that, you know, looks reasonable. And so we can actually use that to predict to some extent where we might find protein coding genes. And introns make that a little bit harder. And we'll talk a little bit more about that when we get to RNA-seq. But the regulatory regions are next to impossible to figure out based on first principles alone, because of course we don't know what, you know, for some random non-coding piece of DNA that's near a gene, whether or not it's important for regulation. And so what we need are techniques that allow us to figure out, you know, what regions are important. But of course, now we have a chicken egg problem, because we don't know what actually binds with those regulatory sequences, and we don't know where they are. So how do you even get started? And so the first set of examples that I want to use are just pointing out that pretty much the different types of things we might care about, the different regulated states, frequently are either promoters, which are defined by being the regions right near the transcription start site. So these are the regions that attract the basal transcription machinery. We might care about regions that we're going to call enhancers, that are more distal elements, meaning elements that are further away from the promoter, but still turn on and off nearby genes. And then we might have other interesting types of DNA, such as insulators, which might block an enhancer from functioning at a promoter in one direction rather than the other. And so these are different examples. And one thing that they all have in common is in all cases, these have proteins that bind to them. And so you can think about this from a biochemical perspective and say, you know, if we just had an approach that allowed us to reasonably easily map where there are high affinity binding sites for proteins on the DNA other than histones, we would be in a position where we would be able to get good guesses at where these different types of elements are. And so, in fact, that's one of the things that molecular biologists and biochemists have worked on, are different ways to isolate nuclei and just say, where are their transcription factors found? And it turns out there's a handful of ways to go about doing this. There are different enzymes you can add, and some of those enzymes are more likely to act at sites where you have proteins bound. And the one that I'm going to focus on first is called DNase. So those of you who remember your biochemistry will know ACE tends to mean you're cutting something, and DNase, therefore, is unsurprisingly an enzyme that cuts DNA. And DNase at high concentrations will completely degrade DNA. But at very low concentration, DNA has specificity and is much more likely to cut DNA that is right next to high affinity binding sites for proteins. And once again, this is not a biochemistry lecture, so we can talk about why that happens. But the bottom line with this is that the DNA that's bound by transcription factors tends to expose DNA in a different way that makes it more susceptible to cutting by DNase. And so you can get so-called DNase hypersensitive regions, which are, if you can go through and map all the sites that DNase will nick most easily, those tend to be protein binding sites. So that's one type of experiment, and as you won't be surprised, given the theme of Wednesday's lecture and today, the way people now map where those sites are is you throw in the enzyme, it makes nicks, and then all you have to do is figure out how to add adapters to those nicked sites and sequence those particular pieces so that you can figure out which regions are DNase hypersensitive. But I want to tell you about another similar way that's actually even easier to do and is kind of amazing. So let's see, I think I have, yeah, sorry, I'm just showing. So we're talking about the transcription factor binding. I'm just keep catching up with my text here. So the idea is that we just talked about DNase-1 hypersensitivity, but that's one of multiple techniques. And one that I'm going to emphasize now is called ATAC-seq. It doesn't matter what ATAC-seq stands for, but let me tell you how it works because it's kind of cool, which is instead of having just nicking your DNA, if you nick DNA, then you have new ends and you have to do molecular biology to locate those ends. But what people have kind of realized over the last decade or so is that it's actually possible to use, instead of a DNA, we can use something called a transposase. And a transposase is a enzyme that inserts DNA into the genome. And so the cool thing is you can actually go through and you can take a transposase enzyme, load it up with known DNA sequences, and throw it into your chromatin. And it'll go, and similar to DNase, it will insert its DNA at accessible regions. But the cool thing is, because we know what that DNA is, we can actually get the transposase to make our sequencing library for us. And so there is an enzyme called TN5, and you'll see it show up again in this lecture because this is a really efficient way in order to insert DNA and allows us to map sites in the genome that are particularly sensitive or particularly exposed, which once again gives us a generic way to try to find protein-bound sites in the genome. I feel like I've been talking for a little while over Zoom without breaking for questions here. So these are a few general ways to look for binding sites in the genome. Does anyone have any questions about this or some of the chromatin content that I talked about earlier?
Matt, I have a more hypothetical question here. A lot of these functions that we are talking about, whether it be sort of cutting at specific sites or sort of adding DNA, could also be done using CRISPR. Is CRISPR being used in this context then?
Yeah, so it's a good question. And I think that that'll help kind of distinguish two different scenarios. So one is we know exactly the site in the genome that we're interested in, and our goal is to try to edit it or target it somehow. And so in that case, CRISPR is very useful technology. In the cases of what I'm showing here, though, generally what's happening is you have your entire three, gigabytes or gigabases of genome or haploid genome, at least in the human case. And we are in a position where we don't know which of those regions are important. So in CRISPR, you would have to have a guide RNA that would go somewhere to cut. And so CRISPR is a really great way to focus on a specific, down to a, one or two base pairs, or at least very few base pairs of region. Whereas these techniques are ways to generally cut throughout the entire genome based completely on, some sort of very generic biochemical property, in this case, transcription factor binding. And so in this case, since we don't yet know prior to these experiments where things are bound, we don't have guide RNAs that can target those. So instead we use generic and non-specific enzymes that bind, and their only specificity has to do with the biochemical properties of the chromatin where it's bound.
Got it. Thank you, Matt.
Great. Other questions? Great. So moving on then, let me just show you what some of this data looks like. So pretty much you get sequencing reads from these types of approaches. And right, the idea is that each one of these approaches, let's take ATAC-seq, so that's the one based on transposition, where if you have a transposon that goes on either side of your site, you sequence across it, and you get sequencing reads throughout the entire genome because you'll randomly insert at some probability anywhere. But the whole point of the biochemical specificity is it's more likely to insert at sites where there are regulatory elements. And so if you get more sites of insertion, you'll get more sequencing reads in those areas. And so essentially you can see these genome browser tracks as just histograms across the entire genome. And usually we're focused in, so if you look at the scale bar here, you'll see we're looking about 100 kb of the genome in this particular figure. So this is a very, very small fraction of the genome. But what you can see at this scale is that you have these different peaks of buildup of reads that happen across the sites that are interpreted as regulated chromatin. And the main purpose of this particular slide is to show that the different techniques that I had you on the last slide tend to agree, even though they use different principles to identify sites of transcription factor binding. They all tend to agree where those sites are, which is reassuring, because if you have multiple independent ways of seeing something and they all agree, it tends to make it less likely that any one artifact is, causing your signals. And so the idea is that in this, 100 kb, if you were trying to figure out where there are likely regulatory sites for some of these genes, these peaks would be good bets for you to study more carefully as a biologist. So once again, this is nice, but we don't really know anything about who's there, right? We just know that some protein appears to be bound there that's causing these various techniques to tell us that there's a site there. I will note that this is the ATAC-seq, which I focused on at the end here. Part of the point of this particular figure is that ATAC-seq can work on very limited cell numbers. And so this is particularly powerful when you have limited material, and that's not surprising given that the transposon or the transposase makes it much easier to prepare the libraries. Okay, so let's talk about what happens. Let's imagine that we actually care about a specific factor. Let's say we have a hypothesis on our last slide. We see that there's a piece of DNA that's bound, and maybe we recognize that that piece of DNA has a consensus sequence for a known transcription factor like MYC, which is a known oncogene, or something like that. And we want to say, okay, we want to test the hypothesis. Does MYC bind specifically at that region? Or let's ask one step more general, where all does MYC bind throughout the genome? Well, the idea here would be we can do a chromatin immunoprecipitation experiment, which is still one of the most common ways to ask this question, which is to say, why don't we take our protein that we want to know where it binds. And what we're going to do is we're going to just cross-link it, meaning we'll use the chemical that's going to actually covalently attach it where it's bound so that we can take a snapshot. So we take our cells, we add a cross-linking reagent, which makes it so our protein is now stuck covalently to the DNA where it's bound in the cell. And then what we can do is break open the cells without having to worry about our protein moving around because it's cross-linked. And we can shear it up into teeny little pieces. And then if we do an immunoprecipitation experiment, meaning we have an antibody that binds to our protein of interest. So in my example, I was saying MYC. So we have an anti-MYC antibody and we pull out MYC. So we do an immunoprecipitation that allows us to mobilize the MYC and wash away everything else. Well, because MYC was covalently attached to pieces of DNA, when we pull out MYC, it's also going to be covalently attached to the DNA where MYC was bound in the cell. And so that's pretty cool because then all we have to do is reverse our cross-links. Oops, I'm getting behind here. We share our chromatin into small pieces, and then we enrich using our target antibody. And once we've done that, we have enriched not just MYC, but also all of the DNA regions where MYC was bound throughout the genome. And then all we have to do is reverse our cross-links and use the exact same library prep that I showed you on Wednesday to go through and sequence. And what will happen is, just like in the last slide for ATAC-seq and DNA hypersensitivity, we can look at the entire genome essentially as histograms where we have a buildup of sequencing reads over all the regions where MYC is bound. So that's pretty cool, and that's called chromatin immunoprecipitation. It's been around since the 1980s, and there's all sorts of follow-ups to approaches such as MIC. Of course, when you get these raw data, you do have to interpret them. So meaning you have to get all the reads, you align them to the genome, and we have to figure out where we actually believe MIC is bound based on comparing our signal to the input. So it's not quite as trivial as just reading it off the raw data. And eventually we need to call peaks so that we can go through and say, okay, here are all the regions that we think MIC significantly binds throughout the genome. So that's kind of the general workflow for a ChIP-seq experiment, both in terms of the wet lab, and then once we have the data, the bioinformatic, really basic analyses. And I believe you'll talk more about that in this course, meaning these types of enrichment analyses. Once again, my role here as someone who goes back and forth between computational approaches and wet lab work is partly to emphasize the fact that there's all sorts of limitations to these approaches, and for you guys to be intelligent consumers of kind of bioinformatic and genomics technologies, you need to realize both wet lab challenges as well as the computational and statistical challenges. And to start with, you can think through every single one of the steps of an experiment like ChIP, and I'm doing this for ChIP, but the truth is you can do this for pretty much any of the applications of the sequencing technology that we're going to talk about. And the idea is essentially you can have questions such as, when you use a reagent like formaldehyde, does it equally cross-link all proteins to the DNA with the same levels? And the answer is no, it doesn't. It depends on the composition of the protein, how tightly it's bound, other issues associated with the sequences. So it's, the cross-linking might not be completely uniform. The question of how good your enrichment is really a question partly of the antibody quality. So how easy is it for you to get a really high quality antibody that only pulls down what you want and nothing else? And then also you can imagine in some cases the accessibility of the epitope. What does that mean? That means if you have a peptide that the antibody has to bind to, and that peptide is buried in the chromatin, it might be harder to pull that particular protein down than a different protein. So it turns out that it's surprisingly rare. Does that matter? And once again, we could talk about the biochemical details and why that turns out not to be quite as big a problem as it sounds like it might be. But either way, all of these are limitations. And then, of course, one has to remember that because of all of these, it means that sometimes the kind of quantitative approaches we tend to use to analyze these data need to be taken with a grain of salt, because a lot of times, because we're not really in a linear range when it comes to the cross-linking or the pull-down efficiency. What that means is we need to be pretty careful about our quantitative analyses of data such as ChIP data. And while it's still useful to do quantitative work and use statistical models, it's worth remembering the limitations of those models and the fact that frequently they rely on premises that are not going to be accurate, and at best are approximations. That doesn't mean, once again, that you shouldn't try, but just that you need to be aware of those limitations. Let's see. So, of course, ChIP has been around since the 80s. Pretty much anything I'm talking about Wednesday or today, there's a gazillion new technologies that I won't have had a time to talk about. And so I try to go deep just occasionally so you get some sense of what those are. And so, for example, with ChIP, generally you're limited by the resolution of how much you shear up the DNA. So if your average DNA is 200 nucleotides, it's hard for you to say within 10 nucleotides of exactly where your transcription factor is binding, because the pieces of DNA you're pulling down are pretty big. And so you get broad ranges, but people have tried to overcome that by using exonucleases to chew up the DNA all the way right up to the binding site, so that you can try to get single nucleotide resolution information about where binding events are happening in the genome, and that's called the ChIP exo experiment. Something that relates back to what we talked about when we were talking about ATAC-seq. People have realized that maybe instead of using sheared up DNA, why don't we leave the DNA intact, but then fuse our antibody to a transposase like TN5? And then the cool thing is that our antibody will actually direct a transposition event right at the binding site. And so there we can use, instead of having to do all of this huge amount of work to prepare a library, we can let the transposase add our adapters for us. And that allows us to get down to very small samples or even single cells. And what you've seen is that if you look over the past even just a few years, these cut and tag and cut and run experiments that were pioneered by Steve Henikoff's lab have really started to pick up steam. And I suspect that you're going to see this continue to be the case, especially for those of you who are interested in single cell genomics type applications, because you really can't do chip style experiments on single cells, whereas a cut and tag offers you a way to get single cell information about localization of factors. And then finally, one thing my lab studies are non-protein factors that bind in the chromatin. And so it turns out there are RNAs that code specific regions of chromatin, including exist, which is a non-coding RNA that inactivates one of the two X chromosomes in female cells in mammals, causing balance of dosage between the sexes. And so I just want to note, not even though there's a protein-centric talk, because that's more common, it is worth noting that there are extensions of things like CHIP2 RNA. And I developed one of these types of approaches as a postdoc, but there are plenty others that have been developed as well. So let me just dive one step deeper into the cut and tag one, just because I think it's pretty cool, which is just the idea, once again, is that we can go through and we can take the chromatin and add in, instead of shearing it up and doing a chip experiment, we can take an antibody and have it direct where a TN5 transposase goes in, and where it goes in, it'll cut and add in this DNA element. And because we preloaded our transposase with a known DNA, we can use sorry, I'm having some screen issues here. We can use the sequencing reads here to actually figure out where the histone modifications or proteins were in the genome. And once again, I'm showing you an example showing that ChIP-seq and cut and tag give us the same answer about where you find these modifications, which once again shouldn't be reassuring to you. If I'm telling you these things work, they should work regardless of which technology you use. So I'm going to do one just polling question and then take some questions. But so let's talk about cut and tag for a second. So we were going through and talking about cut and tag of having some advantages, and this is just partly for me to get a sense of whether people are following or not. The question is, so which are advantages of cut and tag over ChIP-seq, I should say. That should be written in here. So comparing cut and tag to ChIP-seq, which of these things are so all of these are advantages of cut and tag, except for one of them. So please tell me, which one do you think is not an advantage of cut and tag? I'm going to go ahead and end the poll, even if you didn't have a chance to vote, that's okay, and share the results. Okay, so let's see. So it looks like the folks in the class think B should be the answer. Does someone want to unmute and explain why B is the correct answer here? I know 10 of you chose B.
You would still need high affinity antibodies in order to bind to the transposase.
Yeah, right. So the transposase doesn't know where to go. You need an antibody to tell it where to go, right? And so you still need an antibody. And you'll notice that the wording here, I said endogenous proteins without high affinity antibodies. In some cases, people get around this by adding exogenous proteins. So meaning, let's say I care where MYC went, even if I don't have a good antibody for MYC, maybe I could add a tag and express a heterologous version of MYC that has an epitope on it and that would make it easier for me to do cut and tag, even if I didn't have a MYC antibody. So you can play tricks like that, but by and large, you still need antibodies there. However, it definitely simplifies library preparation. The cut and tag, because it simplifies library preparation, it allows you to perform it on very few cells. And then because the transposase can go into the double-stranded DNA without it being sheared, you don't need to have the chromatin shearing step. So therefore, any of the weird artifacts that happen because of needing to shear chromatin aren't issues. Any questions about that before we move on? Okay. Any questions about kind of genome annotation and ChIP-seq more generally, because I'm about to switch gears here. All right. Cool. So I'm going to switch here. So we've been talking about annotation and really been DNA focused, thinking about DNA and the proteins that bind DNA. But of course, DNA is not the only important nucleic acid in the cell. RNA is also an important nucleic acid. And in fact, probably sequencing approaches based on RNA-seq are as important as DNA-seq and therefore deserve a moment of our time. It seems in some ways like it's almost trivially different. They're both nucleic acids and in principle we can sequence them, but it turns out that, what we're doing and, what we want to, how we want to contrast this with DNA-seq, it turns out there's a bunch of differences. So partly there's technical methodology that's different. There are issues with RNA. There's how we do the read mapping. And it turns out that because of the fact that RNA is processed, that can get a little bit more challenging. It turns out that RNA it's not as simple as just being a direct copy of the DNA. And so we can get different isoforms from the same gene. So we'll talk a moment about that. We'll talk about, we won't talk a lot about de novo transcript reconstruction, but hopefully it'll be kind of implicit in what we're talking about. Of course, different RNAs are expressed at different levels, so we'll think a little bit about the sensitivity of sequencing data and what depth we need to see, which regions of the genome are being sequenced. And of course, frequently when we're doing these types of experiments, we care a lot about differences between different samples, so an experimental sample compared to a standard, control sample, and therefore we care a lot about differential expression analysis, which is just what we mean by that. How is it different? And how? Okay, so the RNA-seq workflow itself is fairly straightforward if you already know the DNA one. And the main difference is just we have to get from the RNA to DNA. But once we get into the DNA, then we can use all of the same approaches that we talked about before. So how do you take RNA and get it into DNA? And the answer is, well, the real key point is that there is going to be an enzyme called the reverse transcriptase that's going to reverse transcribe the RNA into DNA. And that's the real key. And once you get to DNA, everything going forward is pretty similar until you get to the data analysis. But so to start with, though, there's a few things we need to talk about that are relevant to RNA-seq that are less of an issue sorry, just making sure I rearrange some of these slides. So trying to make sure. So let's, we'll come back to this in a moment, but the questions are, when we're dealing with the library prep and sequencing, what are the principal challenges that we need to address? And so I think that this slide kind of summarizes what I think the real challenges are. First is, unlike DNA, where there's essentially 2 copies of every piece of the genome, right? I mean, there's of course some repetitive regions. I don't mean to belittle the repetitive DNA, but on the other hand, most of the, genes in your genome, there's exactly 2 copies of them because they're, because we're diploid organisms. Whereas with RNA, some RNAs are expressed at very, very high levels and others are very low levels. So if we want to be able to study the ones at low levels, we're going to have to get tons and tons of reads of all the high level ones just to see an occasional read from a low level RNA. Second is, unlike DNA, RNA is strand-specific, meaning we care whether you made the RNA from the Watson strand or the Crick strand, right? And so we do want to preserve directionality, whereas with DNA, we assume it's double-stranded, and therefore we don't care so much which strand we're sequencing. There's, oopsie, sorry. Thought I had my, I don't quite know. Yeah, well, I should ask someone who knows more how to get my notifications off here. Let's see, the RNA itself, anyone who's worked in a lab with RNA knows RNA degrades much more easily than DNA. And so that has both practical handling considerations, and it's also something to be aware of when you're looking at certain data sets. Sometimes it's hard to compare different samples because they have different levels of RNA degradation, and that can really screw up analyses. So it's not just a theoretical concern, it's a very real practical one. There's this processing, so the splicing of RNA, which means we can't just use the genomic information to guess what our RNA sequence is, because the RNA sequence might be discontinuous with respect to the genomic information, because your exons come together and you cut out giant introns. And then finally, RNA, unlike double-stranded DNA, which the biophysical properties of different parts of the genome vary a bit, but not very much, RNA has hugely different properties because it can have secondary structure. And that can have practical considerations in molecular biology, such as you can block reverse transcriptase with elements such as G quadruplexes. So these are all things that you should be aware of that really distinguish RNA-seq. The other thing that's like super relevant and something that no matter how much you guys work in the wet lab is important to know is that in general, ribosomal RNA is the most abundant RNA in any cell type. And the problem with that is that, you know, if you do RNA sequencing experiments, if 70 to 80% of your sequencing reads end up being over ribosomal RNA, that's a huge waste of money. And it really can suppress your sensitivity of your experiment because you're getting so many ribosomal RNA reads. And so most RNA-seq preps have some way to get rid of the ribosomal RNA, but none of them are perfect. And so when you're talking about wet lab practical constraints that change data sets, depending on what data set you're looking at, people might use different ways to get rid of the ribosomal RNA. And so there's a few different ways to do it. One common way to do it is to actually use biotinylated DNAs that hybridize to your ribosomal RNA. And then that way you can try to pull out all of the ribosomal RNA to get it away from your sample. And so that's one way. So you can deplete it using these types of capture approaches. There's also lots of other ways. There are enzymatic ways where you use enzymatic approaches to specifically degrade ribosomal RNA. And finally, there are approaches that are based on using the fact that that mRNAs have a poly-A tail to only sequence off polyadenylated RNAs, because you're using in that case the fact that that ribosomal RNAs are not polyadenylated. And so I'm going through a whole bunch of these. I clearly am not emphasizing those details except for those of you who are doing these experiments. But the reason I want to emphasize this for the purposes of this lecture is if you're using different data sets, each one of these different approaches can have different effects on biases that will show up in your RNA-seq library. And therefore, it's important to know how the authors of the study did this, even if you're not doing it yourself. Okay, so just to kind of go through one of the most common versions, you can take mRNA, and if you want to get rid of the ribosomal RNA, like I said, frequently people will go through and they'll capture only the polyadenylated RNA with a poly-T oligo. And so that way you'll get rid of all the ribosomal RNA because it's not polyadenylated. When I say all of it, I mean, well, in a biochemical sense, you'll get less of it coming through. Of course, you always get some contamination, but it gets to a reasonable level. And then the idea is what you can do is you can take the RNA fragment and then do cDNA synthesis. And frequently this is done using primers. So it turns out that the reverse transcriptases require primers. And once again, I'm intentionally not including this stuff and just saying it for those of you who care about this level of detail. But the idea is you use primers that have known sequences on them. And you can use all sorts of tricks in order to make cDNA where you know what the two different ends are so that you can get strand specific information. And there's so many different tricks and no one of them is common enough for me to emphasize it, except to say that we do care which end is which because we care which strand the RNA was made from. The idea is we can go through all this and eventually we get back to the same position we were in before, which is that we have double-stranded DNA with known adapters at the end where those adapters have been designed so that we can put them onto an Illumina sequencing instrument, and the YCGA will then send you your reads and you can align them to the genome. Yeah, so before we talk about mapping, does anyone have questions about library prep? And I want to just reiterate that I intentionally went over some of the details just because it gets a little bit arcane and depending on your library prep, each one is so different from each other. I didn't emphasize any given one. All right. Well, so once you get these reads back, I want to emphasize the reads look different than DNA sequencing data. So DNA sequencing data is going to be pretty even throughout the genome, whereas RNA-seq data has this pattern where because RNAs are processed and all the introns are cut out and degraded, the only sequences that are well represented in the RNAs are mostly the exons. So if you look at RNA-seq data, it looks like this, where the gene models below and these thick bars are the exons. And then the areas that are thin in between with the arrows, those are the introns. And when you sequence, what you see is that you get huge amounts of buildup of reads that go over the exons because those are stable and build up, and all the intronic reads, you have a few reads over them because there are RNAs being made, and those are the rare RNA where the intron hasn't been cut out and degraded yet. And that's why you get a few reads here, but most of your reads are going to be over the exons. And of course, the reads themselves, even though they don't show it in these sequencing tracks, some of the reads that are piling up here are reads that actually go across these exonic junctions. So in this case, if this is an intron, you'll actually have reads that go from here and then they hop across intron and go there because, you know, in the RNA, those 2 points are connected. And so what that means is this track doesn't really show it, but you actually have reads that show that these exons are connected to each other covalently. And that turns out to be pretty useful when you're trying to get isoform information. When you're doing these alignments, of course, it becomes a little bit complicated because you have splice sites. So you can use existing annotations. And by annotation, we now mean not only the genome, but also frequently we can go through and know where you have introns and exons. So you know where to look for those discontinuous regions. And so you get these gene annotation files, or frequently GFF or GTF files. For any of you who've done a lot of alignment, you'll know what I'm talking about. And of course, the annotation files actually get to be a little bit complicated because you have to decide what annotation to use, and you find different splice isoforms in different samples, and you never quite know if you want to use more basic or more complicated ones. And so therefore, the annotation files add an extra level of complexity to analysis of RNA-seq data. Of course, even if you don't have annotation files, if you have a genome, at least you can guess what's going on based on the patterns of where your alignment is, and you can try to find de novo splicing using alignment to the genome. And, you know, in the most extreme case, you might not even have that, in which case you can do de novo transcript assembly, just like you do de novo genome assembly, in a manner that I'm not going to go into in depth, but you can kind of guess based on what we talked about on Wednesday, how we did genome assembly using very similar approaches to try to figure out the transcriptome assembly. Any questions about this? Okay. I think some of these things are a little bit complicated, so I'm a little worried that there aren't questions. That being said, hopefully that's because a lot of you are already experienced here, but please do feel free to interrupt me if you think of a question. Okay, I'm not going to talk a huge amount about this, but I do just want to mention that because there are different RNA isoforms, there's a lot of complexity in figuring out, especially from short read data, which isoforms are present and absent in your sample. And if you think about the problem, just imagine that you have 3 different RNAs all from the same gene. You have the red one, the green one, and the blue one. And what you can see is that in some cases they share some exons, and in other cases they differ in certain exons. And the idea is that if you have these 3 different molecules and then you shear it up and you get a bunch of short read sequencing data from these different isoforms that are all in the same cell, you can imagine you have some reads like A, which could map to any of them. So here in this is showing that it's mapping to any of the 3, and we can't tell which of the isoforms it goes to. In B, it might go to the green or blue isoform, but not the red one. So we can kind of say this supports evidence for those 2 and not red. And then we have other ones which are red or blue. So of course you have all sorts of combinations, and every once in a while you'll get a sequencing read that really only maps to a single possible transcript isoform, such as this read D, that is continuous only on that one there. And the idea here is that this is kind of a mess, because if you have this set of reads, trying to figure out which of these is a dominant isoform requires a lot of more complicated statistics, because it's not necessarily clear how many reads of D you should have for every read of E to C to B to A if you have different levels of the blue to green to red. And so this is a real challenge, and something that is an ongoing problem in RNA-seq is coming up with better and better methods to accurately quantify different transcript isoforms. This is just another way to look at that same thing, but to try to motivate why we care. And I probably should have switched these slides, but the idea is pretty much different RNAs in different cells actually have used different exons. And so, for example, this is an example of a transcript where there are 2 different isoforms of it. And if you look in the testes or liver, you find this isoform 3B that includes this exon, but not exon 3A. And if you look in skeletal muscle or heart muscle, you have 3A and not 3B. And I'll remind you, this isn't all just random stuff. 3A and 3B, these are exons. These code for the protein. So this is the difference between maybe having an extra domain or not, or having a regulated phosphorylation site or not. And so the idea here is that the different RNAs that are used are going to matter to the biology. And so we want to actually have RNA sequencing approaches that are going to distinguish between the different transcript isoforms. And that's why all the information about the alignment and quantification of these different approaches matters. What you're seeing in this figure is you have these bars here. These are sometimes called sashimi plots. And the idea is pretty much each one of these bars here connects reads that go from one exon to another. And so the thicker the set of these reads, the more reads there are that actually are found that span these 2 exons. And therefore, if you have a lot of reads spanning them, that's a lot of evidence for those exon-exon junctions, whereas in cases where you have fewer reads, you have thinner, or you completely lack evidence for those exon-exon junctions. All right, that's about all I was going to say about transcript isoforms, I think. Does anyone have any questions about that?
I'm
Cool.
sorry.
So, yeah, go ahead.
I don't quite understand why RNA only has the exon part.
Yeah, okay, great. So let me explain again here, and let me just get an annotation layer going so that we can look at this. So, okay, so pretty much if you're looking at these tracks, right, these once again are histograms of sequencing reads in this region of the genome, and this is exactly where we are in the genome. That's 100 base pairs to give you a sense. And the idea is that if you go through and if you do an experiment where you capture from a population of cells RNAs that are just being made, so if you do something where you just look at the brand new RNA, what you would see is an equal coverage of sequencing reads across this entire region. And that's because as the RNAs are made, a polymerase goes through and the polymerase is transcribing this entire region. And so if you only looked at brand new reads, you would have even coverage just like you do in the genome for any region that's being transcribed. However, what happens is over time, the RNAs that are made from this region actually have exons And those exons are subjected to splicing. So let's imagine that this is exon 3A. And what happens after it's made, and it's actually co-transcriptional, there's machinery called the spliceosome that will come and it will actually cleave out this region that's called the intron. And for those of you who remember, it makes a lariat, and there's there are multiple steps here, and you end up getting a covalent linkage between these two ones. And so just to skip over the data here, that ends up making a covalent link between those two exons and the rest of this RNA here. And then also you get the intronic RNA, and I'm just drawing it as a lariat for those of you who know this is actually what happens. But the key here is this gets rapidly degraded. So this is supposed to be a trash can. There we go. Whatever, you get the point. It gets degraded. And so what happens is this guy is stable. And when I say it's stable, what I really mean is the final transcript with all exons spliced is stable. And this stuff gets degraded very quickly. And so what that means is that at any given time in the cell, you have lots of this stuff. And the only RNA that's found for the introns is the stuff that was just made, because anything that wasn't just made gets degraded. So if you go in, and because these guys are stable, they're going to build up over time. So you're going to have tons and tons of molecules that look like this, that have all the exons, for every one molecule that looks like this guy here. And so therefore, we're going to get a few sequencing reads from this intron. And so you'll have a few random reads there. But mostly, all of your sequencing reads are going to be the reads that correspond to these exons. And so all the reads are either going to be inside an exon, or they're going to go across exons. And the ones that go across exons, I can annotate looking like that. But in reality, those are just reads that are continuous across those exon-exon junctions. And so what ends up happening when you do the mapping is you just get pileups all over all of the exons and very few reads for the introns because they're transient and get degraded. Does that answer your question, Kenyu?
Yes, thank you. So that's just the way the environment of a cell is, right? That's just naturally it is.
Yeah, and I mean, the way you can think about it is RNA sequencing is a steady state measure, or as a measurement of the steady state levels of RNA. And so you have all these different populations of RNA, but the ones that build up are the really stable ones, and the other ones that get degraded. And so every RNA is, its levels are kinetically determined by how quickly it's made minus how quickly it's degraded. So all the stable RNAs, there's a lot more of them given the same rate of synthesis, right? And so there are various ways to look at this. I know you guys have different backgrounds, so I'm just trying to give it different perspectives on it. Cool.
Thank you.
All right, so I think we're doing okay time-wise here. So let me just kind of, oops, I want to clear all my drawings and get out of annotation. Okay, so just kind of going forward with this, RNA-seq is incredibly important. I decided at the last minute to leave out the COVID stuff, but I was reading up as I was preparing for this, how people do, of course, COVID is, or SARS-CoV-2 rather, is a, RNA virus. And so everyone who's doing RNA-seq right now, to look at the different virus variants that are out there, to track the different populations, figure out the virus spread, all of those kind of things are using RNA sequencing technologies. And if you're interested, I'd be happy to send one of the documents that's all the guidance that people use for what technologies to use in order to do RNA-seq of SARS-CoV-2 to study these things. But the point is that all of these choices and technologies are really rapidly developing. You can use them for all sorts of things. So, of course, tracking changes in the virus over time is one that we read about in the newspapers today. But, there's all sorts of different things that people who study splicing, of course, are very interested in RNA-seq, because we can look at these different transcript isoforms. I'm not emphasizing it right here, but differential expression analysis is a huge, huge topic to try to tell, really RNA-seq is one of the best ways to get a snapshot of the biological state of two different samples. So if I perturb a sample, do something like stress it, add a drug, give it a disease mutation with CRISPR or whatever, and you want to say, how does that change the cell? Even if you're not interested in RNA per se, one of the easiest ways to get a global view of the biology of what a cell is doing RNA-seq. So you'll find that in many cases, you'll do comparative RNA sequencing, regardless of whether you care about RNA biology or not. Of course, there's all sorts of cool things RNAs themselves do, and I'm one of the members, a lot of you may know, Yale has for a very long time been the center of a really vibrant RNA community. A lot of other people have started to catch on that RNA is cool, but I would argue that Yale has been very good at that for a long time. We continue to attract a lot of the top RNA researchers in the country to Yale, thanks to our work in studying RNA biology. And there's a lot of really cool RNA species out there that are functional in and of themselves and don't just encode for proteins. And those include large non-coding RNAs, enhancer-related RNAs. You find the entire genome is actually transcribed in most places. I mean, at least 70 or 80% of it is transcribed at some point. And so there's all sorts of interesting questions to ask about the RNAs that are made from non-genic regions. Of course, there's all sorts of interesting things to say about translation. And in fact, the ribosome itself is a ribozyme, meaning the RNA is largely, or the ribosome is largely made of RNA. So, labs like Susan Berserga's lab here, uses techniques like RNA-seq to study that. There's all sorts of interesting things if you think about the different RNAs that get made from the paternal maternal allele. So the gene that came from your mom and your dad, they have some differences like single nucleotide polymorphisms, and you can use RNA-seq to figure out if, if both of them are being used or just one or just the other. So there's allele specific expression. If you look to see which SNPs are in the RNA compared to which ones are in the genome, you can get cool information there. And there's all sorts of applications of that. If you're a lab like mine, we care a lot about RNA metabolism. So we've been developing tons of techniques using metabolic labeling to measure RNA half-lives and decay. So really questions associated about the dynamics of the transcriptome and how it changes over time. You can use a lot of these RNA-seq approaches to ask questions such as where do proteins bind to RNA? So RNAs are trafficked around in the cell, and whether they're translated or not and degraded or not are determined a lot by proteins that bind them. So just like CHIP for chromatin immunoprecipitation to look at proteins binding to DNA, there are similar approaches to look at proteins binding to RNA, and those have names like CLIP and RIP, and once again, there's tons of names, and sometimes the names don't even mean that much because everyone invents a new technology that's based on the last and gives it a brand new name. But the key is to understand the principles that these things can be done. And then finally, we can look at all sorts of interesting, other interactions between the genomics, being what's in the genome, and the RNA biology, including there are a number of really exciting approaches where people are actually using antisex technologies or even drugs that target RNAs to cure diseases, or at least to try to with therapeutics. And there are a few impressive recent examples. So this is a really hot area to think about RNA in that way. And of course, RNA sequencing is very central to that field. So I think, just to kind of wrap up, I think the one really big example that's worth discussing, with this class before concluding is, single cell RNA-seq, which is frequently abbreviated as cRNA-seq. And the idea here is pretty simple that, every time we're doing bulk biochemistry on mixture tissues or cell lines, minimally, even with cell lines, there are different stages of the cell cycle frequently if you don't synchronize them. And so each cell really has its own biology going on. And that's even more so true in an animal, right? And especially in immune cells and things like that, we might care a lot about individual cells. And a lot of the techniques that were originally developed, you really need, sometimes thousands to tens of thousands to even millions or billions of cells to successfully do some of the techniques I've talked about. But there's a realization that those techniques need to be really pushed down to the point where you can ask questions about genome changes in individual cells, RNA-seq changes in individual cells, and chromatin annotations in individual cells. So I've already told you a little bit about that for techniques like cut and tag, but what you can see is that, if you have diverse RNAs, the idea is that if you can do single cell RNA sequencing, then you can actually go through and take cells and actually figure out different properties of the cells, either throughout the cell cycle or even throughout development. And so there's all sorts of really beautiful ways to kind of pseudo-type cells based on their RNA-seq profiles to figure out developmental lineages of which genes come on and which waves during development or cancer or disease. And so this is a really exciting direction forward, and has, it's been around now for, well over half a decade, let's say. So, these are starting to become mature and increasingly common in a lot of the papers that you guys might read. And I think some of you might already be pursuing these. The techniques are based on, just like everything else in this class, I recognize you can't read this. This is way too small. I have the reference here, and this is actually an old reference. I still find this to be one of the easiest ones to kind of talk about the different types of sequencing that are out there, but the basic ideas are various ways to capture single cells and to sequence the RNA and get the information from them so that we can figure out what RNAs are in which cells. And single cell sequencing has all sorts of specific challenges that are different from other RNA-seq approaches that I don't have time to go into, but I just want to kind of highlight that hopefully the types of things and considerations we've talked about Wednesday and today will help kind of at least give you a framework for thinking about what the challenges are as you get into each new technology as it's developed. So with that, I'd just like to wrap up and just remind you what I hope I've accomplished over these two lectures. The first one on Wednesday was really to give you a sense of just sequencing technology and how sequencing DNA works, and hopefully inspire you to think about those data as not black box, but rather things that can be understood in terms of molecular biology and how the sequencers actually function, and that you would feel comfortable going into your FASTQ files and thinking about their details. And then once we talked about how you can do that and actually can sequence DNA, today we talked about a gazillion different ways that we can use that sequencing technology to learn about biology, everything from developmental biology, most recently in the single cells, pseudo trajectories, down to how cells themselves annotate their genomes. And so with that, I hope I've scratch the surface enough of this that you'll feel some empowerment as you're reading genomics papers to think critically about the technologies people are using and recognize that it really is worth engaging both the wet lab, details and the materials and methods, and also the computational parts. And I hope you're willing to kind of look at those as being a package that all comes together to get a better understanding of genomics and biology. So with that, I'll wrap up and take any questions you guys have. All right. Well, I hope the no questions is not a sign of you guys being lost, but rather, of course, if you do have questions, feel free to email me, and enjoy the other modules and learning about all the data and modeling and mining you're going to learn in this class.